Male internal genitalia, detailed overview. The male internal genitalia consist of organs and structures located inside the body that play a vital role in sperm production, hormone secretion, and the transport of sperm during ejaculation. These organs work in concert to ensure male fertility and the ability to reproduce. The key components of the male internal genitalia include the testes, epididymis, vas deferens, seminal vesicles, prostate gland, and bulbourethral glands. Primero-testes, testiculos. The testes are the primary male reproductive organs responsible for producing sperm, spermatogenesis, and the male hormone testosterone. They are housed in the scrotum, which serves to maintain a temperature slightly cooler than the body's internal temperature, ideal for sperm production. The testes are essential for both fertility and sexual function. Anatomy of the testes. Structure. The testes are oval-shaped, and each testis is about 4 to 5 centimeters long and 2 to 3 centimeters wide. The surface of the testes is covered by a protective fibrous membrane called the tunica albuginea. Seminiferous tubules. The testes contain tightly coiled seminiferous tubules, where spermatogenesis occurs. These tubules are responsible for producing sperm cells. Leydig cells. Located in the interstitial tissue between the seminiferous tubules, Leydig cells produce the hormone testosterone in response to stimulation by luteinizing hormone, LH, from the pituitary gland. Sertoli cells. These cells, found within the seminiferous tubules, provide structural support to developing sperm cells, nourish them, and create a blood-testis barrier that protects sperm from immune system attacks. Function of the testes. Sperm production, spermatogenesis. The testes produce sperm from spermatogonia, stem cells, through a process that involves meiosis and differentiation into mature sperm cells. Testosterone production. Testosterone is vital for the development of male secondary sexual characteristics, e.g., deep voice, facial hair, muscle mass, and the regulation of spermatogenesis. Second, epididymis. The epididymis is a coiled tube located on the back of each testis and plays a critical role in the maturation and storage of sperm. It serves as a passageway for sperm from the testes to the vas deferens. Anatomy of the epididymis. Structure. The epididymis is divided into 3 parts. Head, caput, the upper part of the epididymis, where sperm first enter after being produced in the seminiferous tubules. Body, corpus, the central portion of the epididymis where sperm continue to mature. Tail, cauda, the lower part of the epididymis, where mature sperm are stored until ejaculation. Function of the epididymis. Sperm. Maturation. Sperm entering the epididymis from the seminiferous tubules are immature and unable to fertilize an egg. As they pass through the epididymis, they undergo maturation, acquiring the ability to swim and fertilize an egg. Sperm storage. The tail of the epididymis stores sperm for up to several weeks, releasing them during ejaculation. Drite vas deferens, ductus deferens. The vas deferens are muscular tubes that carry mature sperm from the epididymis to the urethra during ejaculation. Anatomy of the vas deferens. Structure. There are 2 vas deferens, one on each side of the body. Each vas deferens is about 30 to 35 centimeters long and extends from the epididymis into the pelvic cavity. The vas deferens passes through the inguinal canal, a passageway in the abdominal wall, and enters the pelvis, where it eventually joins with the seminal vesicle to form the ejaculatory duct. Function of the vas deferens. Sperm transport. The vas deferens transports sperm from the epididymis to the ejaculatory ducts, which leads to the urethra. Muscle contraction. During ejaculation, smooth muscle contractions of the vas deferens propel sperm forward. Fourth, seminal vesicles. The seminal vesicles are paired glands located behind the bladder. These glands are responsible for producing a significant portion of the fluid that makes up semen. Anatomy of the seminal vesicles. Structure. Each seminal vesicle is about 5 centimeters in length and is located behind the bladder and in front of the rectum. The seminal vesicles secrete a thick, yellowish fluid that is rich in fructose, a sugar that provides energy to sperm. Seminal vesicle ducts. Each seminal vesicle connects to a vas deferens. forming the ejaculatory duct that passes through the prostate gland and empties into the urethra. Function of the seminal vesicles. Production of seminal fluid. The seminal vesicles produce about 60 to 70% of the total volume of semen. This fluid contains fructose, prostaglandins, which help in sperm motility, and other proteins that nourish and protect sperm. Contribution to ejaculation. The fluid from the seminal vesicles is released into the urethra during ejaculation, mixing with sperm from the vas deferens to form semen. Fifth, prostate gland. The prostate gland is a small, walnut-sized gland located just below the bladder and surrounding the urethra. It plays a key role in the production of seminal fluid and contributes to sperm health and motility. Anatomy of the prostate gland. Structure. The prostate is a muscular gland that surrounds the first part of the urethra, prostatic urethra. It consists of glandular tissue that secretes fluids and smooth muscle that aids in ejaculation. The prostate has two main parts, the central zone, which surrounds the ejaculatory ducts, and the peripheral zone, which produces most of the prostatic fluid. Function of the prostate gland. Production of prostatic fluid. The prostate gland produces a thin, milky fluid that is slightly alkaline and helps neutralize the acidic environment of the female reproductive tract. This fluid accounts for about 20 to 30% of semen volume. Semen activation. The enzymes in prostatic fluid, such as prostate-specific antigen or PSA, help activate sperm and increase their motility. Facilitating ejaculation. The prostate gland contracts during ejaculation, helping propel semen through the urethra. 6 bulbourethral glands, Cowper's glands. The bulbourethral glands are small, pea-sized glands located beneath the prostate gland. They contribute to the fluid component of semen. Anatomy of the bulbourethral glands. Structure. These glands are located on either side of the urethra, near the base of the penis. They are typically about the size of a small pea. Ducts. Each gland has a small duct that opens into the spongy urethra just below the prostate. Function of the bulbourethral glands. Pre-ejaculate fluid. The bulbourethral glands secrete a clear, mucus-like fluid, often referred to as pre-ejaculate, prior to ejaculation. This fluid helps lubricate the urethra and neutralizes any acidic urine residues that may remain, providing a more favorable environment for sperm. Lubrication. The secretion from the bulbourethral glands also provides some lubrication during sexual intercourse. 7th, urethra. The urethra serves as a dual-purpose conduit. It carries urine from the bladder, and during ejaculation, it serves as the passage for semen from the male reproductive system. Anatomy of the urethra. Structure. The male urethra is approximately 18 to 20 centimeters long and is divided into 3 sections. Prostatic urethra, the portion of the urethra that passes through the prostate gland. Membranous urethra, the shortest section which passes through the pelvic floor muscles. Spongy urethra, penile urethra, the longest section that runs the length of the penis to the external meatus. Function of the urethra. Urine passage. The urethra allows urine to flow from the bladder during urination. Semen passage. During sexual arousal, the urethra becomes the pathway for semen to be expelled from the body during ejaculation. In conclusion, the male internal genitalia are essential for sperm production, maturation, and transport, as well as for the production of seminal fluid. The testes are responsible for sperm and testosterone production, while the epididymis, vas deferens, seminal vesicles, prostate, and bulbourethral glands contribute to the storage, maturation,